Oh, sure. Yes, please.
So a little announcement. So you might have noticed that today is a holiday in Canada. It's the Truth and Reconciliation Day. And this is used to memorize or to remember all the injustice which was done to the native people of Canada over the years. It was initiated, I think maybe 3 or 4 years ago, so it's relatively new, through the discovery of all the graveyards on the residential schools. So I don't know if you've heard about this or followed this in the media, but there have been lots of residential schools all over Canada where native children would be gathered, taken away from the families, and educated in religion and Western beliefs. And many of them died actually there, and they were buried at the residential sites. And nowadays people find all these graveyards, which is really terrible. So today, if you go through the city of Toronto, you will see many people in orange. So we only have one here, unfortunately, but orange is like the symbol for this day. And you will also see native people in the parks doing their dances or doing their spiritual things. I think also at City Hall, there's a big event today. If you want to check it out, you can go there. Okay, thank you.
Thank you so much. That's great to know. So next up is Jeffrey West from the Moffitt Cancer Center, who's going to tell us about leukemia and machine learning.
Awesome. Yeah, thank you. The goal of my talk is kind of twofold for today. First of all, I want to talk about some methods of combining mechanistic modeling, which we're all very familiar with, and combining that with machine learning techniques. And so this is all new work from my group, and I'd love feedback, and you feel free to ask questions throughout. And then the second goal of my talk is to learn something about ALL leukemia. In particular, we're going to be talking about cell fate transitions, in other words, plasticity. So I'll start with the clinical background, and the easiest way that I can explain the biology is through the lens of something called the stem cell hypothesis. And the stem cell hypothesis will differentiate acute myeloid leukemia, so AML, where researchers for a long time were focused on identifying what is a stem cell, which is capable of transplanting leukemia between mice, and it's capable of self-renewal and thus capable of chemo resistance. The search for these ostensible stem cells is more difficult for a disease like ALL. In other words, we have different stem cells and progenitor or differentiated cells, which can all transplant leukemia between mice. And so what our hypothesis here is that the cell state transitions between these different cell types is much more important in ALL than it might be in AML. But how can we quantify the transition rates between these cell states? That's the goal of this project today. So we have this sort of normal lineage process of B-cell development, and we have some biomarkers which we can measure those cell types in patients. And so we're going to sort of coarse grain this cell flow structure into 4 cell types that I'm going to be talking about for the rest of the talk today. And this purple cell type we'll just call stem cells, which make progenitors in slightly less purple, and hematogones, and then B cells, which are the most differentiated cell. So obviously in ALL, this process is disrupted, and there's some transitions that occur that wouldn't normally occur in the healthy individuals. So this is our data set, right? So to give you some background about the clinical setting that we're looking at, patients come in and they're diagnosed with B-cell precursor ALL, and they're given induction chemotherapy for 4 to 6 weeks. And then they're assessed with if they have any evidence of remaining disease. And if they're positive, they may get an intensification of their therapy. And if they're negative for MRD or minimal residual disease, then they may get some consolidation therapy and later some maintenance therapy. And we have basically information about those 4 cell types at the start of their diagnosis and at the end after they may relapse. Okay, so this is the data set in one slide. I know this is like a very busy slide, and you don't need to see or make out all of the details, but I just want to illustrate kind of what we have and how heterogeneous this data set is. So I'll go through all of the rows just to show you what information we have. So every patient is a single column. And so I don't remember how many patients, maybe 50 patients we have. And we have their age, which is the histogram along the top here. And then each patient has a unique identifier, and we know their sex, male or female. And then we have something called specimen. So we have basically a peripheral blood draw or a bone marrow sample. And sometimes we have both on a single patient. And then we have some genetic status, which I'll talk about in a second, BCR-ABL1 state. And then we have their outcomes. Are they positive for minimal residual disease? And are they positive for relapse after 3 years? And then we have certain genes of interest as well. And the circles down here are basically the prevalence of each of those 4 cell types that I talked about in the beginning. And the most important sort of distinction of separating and stratifying these patients is this BCR-ABL1 Philadelphia translocation. So this is a genetic alteration that's known to be associated with inferior outcomes for patients. And so you can already see some trends in this data, right? So if we look at this cell states given by flow cytometry, you see that these BCR-ABL1 patients typically have larger circles on that first row, which is the stem state. And then the negative patients have larger circles maybe in the middle rows, right? If I show that data in a different way, all I'm saying is that BCR-ABL1 positive patients have more stem cells. And the negative patients have more of the other kinds of cells, the progenitor or more differentiated cells. And so again, I already said this before, but my hypothesis is that stem cells are going to drive relapse and disease dynamics in BCR-ABL1 positive patients. Those are the more aggressive patients. But there's a subset of patients that are BCR-ABL1 negative where we think it's more of a plasticity-driven disease dynamics. So this is our setup, is we have sort of 2 different groups, and we think one is stem-driven and one is plasticity-driven. So we want to see what's the rate of transition of changing those cell types within a patient between those 4 different cell types. Please, yeah, of course.
So exactly where do you see in the negative guys the cell state transitions in the data? I'm sorry.
Yeah, good question. So far, we don't know what the rate of transitions are between patients. We only have the prevalence of each cell type. So the goal of my talk today is to define that rate of transition, which we can't measure in patients right now. So great question. So this is just giving you a sense of where we're going with the data. And in order to estimate those transitions between cell states, we're going to basically try to first predict which of those patients are more aggressive based on the clinical information that we do have currently. So we're going to look at predicting BCR-ABL1 status and MRD status based only on those 4 cell states. And then we're going to revisit it by creating a mechanistic model to define the transition between those 4 states and make another prediction using that information in these rows. Okay, so this is my first machine learning model of the talk, and we're going to use something called linear discriminant analysis, which is similar conceptually to principal component analysis, but rather than separate the variants, it explicitly attempts to model the difference between 2 classes. And we have BCR-ABL1 positive and negative, right? And so it separates those two patients based on just the number of each cell type that we have, those 4 cell types. And if we draw a line through the data, then we can sort of predict that the negative patients are below the line and the positive patients are above the line. So it's fairly separable, this data set. In fact, we get an accuracy of 84%. And we can also predict MRD status, minimal residual disease. It's a little less accurate because this is making a prediction forward in time, 4 to 6 weeks later after that induction chemotherapy, using only the information given at diagnosis. Okay, so this is only like my 3rd slide, and I've already showed you a result, but I want to sort of take a step back and say, what did we learn from this result? We can make an accurate prediction with the data that we were given, but let's talk a little bit about philosophy of modeling. At Moffitt, we often talk about hypothesis generating models versus hypothesis testing models. So hypothesis testing model is what I just did. You begin with the data and you build a model. It could be a black box. We don't know what's going in there, or it could be more descriptive than that. But basically, we're aiming for some prediction given some data set inputs. In contrast to that, we could do hypothesis generating, which starts with logic rather than starting with the data. So we logically write down some set of assumptions, and then we translate those through the model to arrive at some conclusions with or without data. And I like this quote because it sort of says that no matter what your model is, if it's a black box or if it's something like an ODE that's written more explicitly, that we deduce logically what to write down, either way, we start with a set of assumptions and we arrive at a set of conclusions. So my goal today is to sort of take our machine learning black box and open it up and figure out what set of assumptions is driving those predictable outcomes of that 84% and 70% that we were able to sort of predict, okay? Makes sense where we're going. And so to do that, I want to use something that I recently learned about, mechanistic learning. So there's 2 really cool review papers on this, the one from 2020 and one from this year, that talk about combining machine learning with mechanistic modeling. And I think I don't need to convince you all that each has their own advantages. Machine learning deals with big data to make accurate predictions, and mechanistic modeling usually works with low dimensional data, but they provide mechanistic insight, especially in longitudinal systems. And combining these 2 is not really that straightforward, right? So especially the second review gives some ideas on how to combine them, whether in parallel, answering 2 different aspects of the different question, or in sequence. And so today what we're going to do is basically sequential mechanistic learning, combining ML and mechanistic modeling. Okay, so the first section, before that, I'll give you sort of an outline of today's talk, which is based on my title. So mechanistic learning reveals the reciprocal cell-fate transitions that drive disease progression in BLL. So what I just told you is we're going to do what's called sequential mechanistic learning. So what we do is we start with the clinical features, which I just explained to you. And then we build a mechanistic model. And this is what Eduardo was asking is, how do you know the cell state transitions? Well, we're going to derive those from our mechanistic model. And then after that, we're going to feed those into a machine learning model to make a prediction. We already fed in these clinical features to make a prediction, and it worked fairly well, right? But what we want to do is iterate so that we can reduce the list of parameters required to feed into that machine learning model so that we get 2 important things. We want to retain the predictive accuracy of that black box, but we also want to increase our interpretability of what's actually going on inside of that black box. So let's begin with the mechanistic modeling. And here I like putting these little quotes in the interlude slides, and you can see a little bit of my sense of humor with this Albert Einstein quote, if you want to know the future, look at the past. We're going to be talking about Markov chains, which are memoryless processes. So I guess Einstein never worked with Markov chains because he would have said, if you want to know the future, look at the present, since Markov models are only determined by the present state of the system. Sorry for the nerdy joke. Okay, so what is a Markov chain model? You start with a state vector. Here we have those same 4 cell types, X1, 2, 3, and 4. And we can view those as a probability of a single cell existing in each of those states. So if we have a single stem cell in the dish, then our state vector would be 1, 0, 0, 0. In other words, we have a 100% probability that that cell is a stem cell. And we want to look forward in time and see what's the probability of that stem cell differentiating into these different cell types. To do that, we have a transition matrix where you have the rows determining the transition from a particular state, the row, to a particular state, the column. So in other words, that stem cell, you look at row 1, and it has 83% chance of remaining a stem cell in the next iteration, and 8% chance of transitioning to that second state. And so mathematically, you just multiply the state vector times the transition matrix, and each update is equivalent to taking the power of that Markov transition matrix. And if you take the power to infinity, right, then we know the steady state distribution of cells based on that particular transition matrix. And what we're assuming is that we actually know this distribution in a patient. So we're going to assume that each patient is at steady state at diagnosis, and we want to determine the Markov transition matrix associated with that steady state value. And to do that, we're going to use the algorithm that my PhD supervisor, Paul Newton, developed with one of his students, Jeremy Mason, to estimate Markov transition entries. They were looking at the distribution of metastasis, but here we're looking at ALL. And basically what you do is you start with a matrix and you iterate somehow to get an ensemble of matrices that all match that steady state value. So I'm happy to go into more detail, but I'm going to kind of skim over this. But essentially what you do is you sort of start with a guess, and then you iterate and you look at your current matrix and steady state value and the target from the patient, and you see how close they are together. Hopefully they're exactly matching, exactly equal, but in reality, we define some residual error between our current iteration of the algorithm and that where we're aiming for the target V sub t. In practice, we just iterate until some acceptable tolerance, and this is what it kind of looks like. You start with a guess, and the guess is important as well, and then you end up with some convergence so that we match our steady state. But how do we update that each time in the algorithm? Well, as follows. We take that residual error at every step in the algorithm, and then where the error is the highest, we pick the column according to that transition matrix, and then we pick the column of the minimum error, and then we we decrease the element in the max error, and we increase the element in the min error. And you can see that during the training process, we do converge to residual error approaching a very low acceptable tolerance. And our initial guess has an associated steady state value for that vector. But then after a while, the target matches the trained. And then we repeat that for each patient, and we get something like this. Okay, so this is my diagram. So this is important to sort of see how we're visualizing these cell state transitions. We have 4 states. If I start in state 1, you can see this band sort of collapses back on itself. That means there's a huge probability that a cell in state 1 remains in state 1. Well, it makes sense because you can see this 83% here. That band will be 83% of this chord here. There is an 8% probability that we transition to state 2, which is like this right here, and then to state 3, which is right here, so 9%. So they're ordered in rank order of how likely it is to go to each state. Sure. Yeah.
Is there a unique matrix?
Great question. There's not.
It's not.
Yeah. It's a huge space.
So why picking a
Yes, great question. So that's in the next slide. What we do is we create an ensemble of Markov matrices, and then what we have is a sort of an underlying probability distribution for each element. And so all of these in the ensemble exactly match, or within some tolerance match the steady state target vector. And we have an ensemble associated with a mean and variance for each of those individual transition rate parameters. The details here are important, especially with what matrix you start with. And there is some biologically reasonable starting point where we start with basically the stem cell state row, starting with the distribution of cell types in normal healthy individuals. In other words, we assume that the stem state is sort of the smallest Euclidean distance to normal healthy functioning B cells. And then we iterate away from that initial condition. But again, we need to verify the exact transition rates that if we want to actually rely on these for knowing particular values in this transition matrix, right? But yeah, so great question. So basically what we get is a probability distribution associated with a single patient, which is many possible values, a matrix, many possible matrices associated with a particular patient. And for the rest of the talk, what I'm going to take is just the mean value for that patient from those 1,000 repetitions, and then make predictions based on that. So we've sort of removed any sort of outliers and looked at the most obvious examples here. Okay, so what am I going to do with that information? Oh, go ahead. Yeah.
In the approximation that you do afterwards, you're assuming that all the entries are independent? Because I guess you have some constraint.
Right. So are you saying all, if I'm assuming all values M1 to M44 are independent?
Yeah.
I'm trying to think.
So it has to be stochastic.
Right. So yeah, it has to be row stochastic. So they have to sum, each row has to sum to 1, right? So that is one assumption. As far as independence, the algorithm itself retains row stochasticity at every single step. Yeah. So I'm not sure if that answers your question, but yeah.
But when you start to distribute, I understand that which markers you do want the hypothesis of reserve. You understand that on the next slide when you say that each end of the markers in the
Yeah.
distribution is a linear variance, but then you still need to preserve the hypothesis of the markers.
You're right. Yeah. So if we take the mean, then you may not retain row stochasticity. I agree with you. So practically speaking, I don't know if there's a better way to do this, but I take the mean and then I normalize by row stochasticity. And then I check if it's close enough to the target vector, if I've changed the steady state value again. So yeah, very good catch. I'm impressed with that question. Awesome. So yeah, it may not be a reasonable approach depending on if you change your steady state vector during that normalization step as well. You could sort of take like the median matrix or something. I don't know. There's maybe other ways to do it. Right.
It's a complex set. So
Yeah.
if
Yeah.
you do complexification and everything at the same time, you should be okay.
Yeah, that's a good idea. Yeah. I mean, that's a good point. Yeah. Okay, good questions. So now that we have a set of transition matrix for each particular patient, we're going to sort of use that information in order to make some predictions about the biology. So here's what I had before. So the same sort of data set of those 4 cell types. And here's what we get, which is what you were just asking about, the average matrix for each particular patient. So each patient is a column here. And if you look, I'm not sure, do you see any trends? Like it's kind of difficult to see. It's like it's almost like we made our data set worse, right? It's like more noisy and it's difficult to see any trends between like BCR-ABL1 positive and negative patients. Yeah, so what we did is just as a first step, we used this tool called Morpheus, which is normally used for genetic data, but it provides some classification features. And so we took every one of these 16 Markov transition rates and we classified them to see which are the most relevant to each, or the closest Euclidean distance to one another. And so I haven't changed the columns at all here. So the patient data is exactly the same. I've just rearranged all of the rows and see which are the most closely related row. And it just happens to be, without exception, that the rows cluster according to the incoming transition rates for each of these 4 cell types. And so that's kind of a profound result here is, another way of saying that is not every single transition rate provides independent predictive value. If we look at one transition rate, it's coupled to another transition rate. And the coupling is the transition rates into each state are the most similar to one another. So what I'm going to do is I'm going to try to reduce the dimensionality of this problem by looking at the structure of the matrix rather than any individual value itself, right? So there's something structurally about these transition matrix which describes these patient dynamics very well. And to do that dimensionality reduction, I'm going to use something that we call reciprocity, and it has 2 components. It's the relationship between the outgoing states or outgoing transitions for a particular state and the incoming transitions for a particular state. So if we look at just the stem state here, and we want to look at sort of all of the outgoing, so this doesn't have very many outgoing, right? And then we want to compare that to all of the incoming into that state. So this particular BCR-ABL1 positive patients has a lot of incoming to stem and not very much outgoing. And the negative patients has more outgoing, but not very much incoming. And that corresponds to, we have this R1 out parameter and R1 in parameter. And we see sort of the opposite effect here. And we repeat that, or this is a visualization of just coloring only the ones connected to the stem state, right? This top left state. And you can see very visually that there are very strikingly different relationships between the stem state. But we find the reciprocity components for all 4 of those states. And then we go back to our data set. And now rather than plotting the Markov transition matrices, I'm plotting just those reciprocity components. So we have 4 states and we have in and out of each 4th state. So we have 8 parameters now. And they describe sort of the structure of our matrix. And what's really interesting, this is sort of like my favorite part of this talk here, is Sadig, my postdoc, had this really great idea to look at the quality of representation of all 8 of these parameters. So we do a PCA, and then we look at the contribution of each of those parameters to the first principal component. And then if I only look at those 2 parameters and I block out the others, now instantly you can sort of see a relationship coming back that sort of separates BCR-ABL1 positive and negative patients, and in particular, very separable for remission patients. Let me sort of show that on the diagram I showed in the last slide. And you can see that typically these BCR-ABL1 positive patients have a low out and a high in, right back to that stem state, and the opposite for the remission patients. So let me zoom in on that a little bit. And the 2 remaining parameters are just the reciprocity components only for the stem state. So these 2, R1 in and R1 out. So even though I allowed for these others to sort of contribute to the PCA is telling me these are the only 2 parameters that are important in our data set. And if I rank order by high R1 in and high and low R1 out, then you can see that the patients cluster according to their genetic subtype. So this is sort of reducing our information to the most important features of our model parameters from our mechanistic modeling in order to show some accurate predictions. And the answer to this question, does the structure of the Markov transition matrix correspond to BCR-ABL1 status? It's very convincing. So the BCR-ABL1 positives have a high in coming, and BCR-ABL1 like has almost as high, and negative has lower, and remission has the lowest. And so this high one R in sort of corresponds to disease severity. And then R1 out inversely corresponds to disease severity. And we see the same trend hold for minimal residual disease as well. And what's really cool is we can go back to sort of our LDA machine learning method, which was in some sense a black box, right? And this is what I showed before, is just using the flow cytometry data of those 4 cell types, we get some baseline accuracy, but we don't really necessarily know why that's happening. But if I retrain the LDA using only those 2 parameters now, we essentially retain the same amount of accuracy, but we have an exact idea of what biologically is actually causing these patients to cluster. Yeah, go ahead. It does not. So great question. It represents the transition rates after everything is all said and done, right? So including proliferation and plasticity and death due to drug and everything, right? So it's a simplification in that sense. So it would be difficult to tease out if these were dividing at different rates, but the net outcome, right, is just where we end up. Yeah.
So the condition rates that we interpret
Yeah.
are
Exactly right. Yeah. Yeah. Great question. Great question. And so now, yeah, I think that's actually.
May I ask
Go
a
ahead.
question?
Sure, sure. Oh, are you
yeah,
asking me to repeat the question?
Sorry, yeah.
Sorry, yes, I should get in the habit of repeating the question. So the question was, do we account in the Markov transition matrix for cell proliferation rates? And the answer is no, we don't know the difference in each of those 4 cell types proliferation rates. We only know the net outcome of how many cells end up in each of those 4 compartments. So,
to be more precise, it's not how many cells, but the proportion.
The proportion, exactly,
That's
yes.
yeah.
Right. Yeah, so it's normalized. So we don't know how many cells there are in each patient either. Yeah. I don't have it. Maybe we do, but I'm not using it. Yeah. Yeah, good question.
Sorry, may I ask a question? Do you hear me?
Oh, sure. Yeah. If you have a question
Yeah,
online.
okay,
ahead.
yeah. When you say that LDA a black box, what do you really mean? Because with LDA, we can have linear combination
Yeah.
of one of the features. So it may be, we can explain the impact of the cell type to this linear combination. Somehow it's not really black box like the deep learning or other methods.
It's a pretty nice black box, yeah, because it is a linear model, right? So we would be able to assess the contribution of each of those input parameters in a different way than I've done. So I definitely grant you that point. There's no reason why we would have had to use an LDA. We could use something more complicated or different type of machine learning. So the point here is less about the exact machine learning method and more about using whatever machine learning method you're already using, and then trying to identify what's driving that in your predictive data. So great point. Yeah.
Okay, thank you.
Go ahead. Yeah.
Did you do some testing to see if there was much effect of the convergence of Markov chains starting from different initial conditions?
Yeah. So the question is, does the Markov training process converge with different initial conditions? And yes, it will almost always converge. Yeah, I don't have much analysis on that. So as far as why it converges or this and that, but the choice of initial condition is extremely important. Yeah. And so what we try to do is choose the initial condition to most closely mimic healthy individuals. And so what we're implicitly saying is that the transition matrix is sort of the closest departure from healthy, right? Yeah. Was there another question? Yeah, they are highly correlated. So we could probably repeat this with only using one parameter, but I feel like it's more intuitive looking at both. Yeah, but in the Markov chain, they're not necessarily correlated. They could be independent for sure. Yeah. There is one caveat to that, which we'll get to at the end. So another great question of whether or not these two parameters are correlated. Is it what? Sorry. Good question. I'm not sure. So sorry. Yeah, I'm not sure. So yeah, let's come back to that. Good question though. Okay, so this is basically my pipeline. We ended up with two parameters. And what I want to do next is sort of do something interesting with those two parameters in the remaining time that we have. So to build intuition, what we like to do is build another math model, right? So this is not really phenomenological learning, but I am going to build a phenomenological model using those two parameters. So we basically learned that we have stem cells and transitions in and out. So I think we can sort of reduce our system to two states, right? You have stem and non-stem, and we can combine those into two different states where chemotherapy targets one of those states, which is not resistant, right? So the stem is the resistant phenotype. And what we know from the data is what I just told you is that these R1, R out and R in are either high or low, depending on if you're BCR-ABL1 positive or negative. And what I didn't really mention before is that remission, you need a R1 that's approximately 0. And then we know some relationship between how likely it is to have stem or non-stem at diagnosis. And so we write down sort of an obvious first math model. I just say logistic growth with the same growth rate, because like Jasmine was asking, do we know the difference in the growth rate? We don't. So let's just assume they're the same for now. And then all that's different is the cell state switching between these stem and non-stem compartments. And then we add a drug term to the non-stem. And what's really cool is it sort of recapitulates our data set with this very simple model. If I have a high R1 in, that means the stem cell takes over at a high proportion when we have BCR-ABL1 positive patients. If I flip the magnitude of these two, then the non-stem population takes over. So this matches our data. And then if I look at the remission patients, the R1 in is basically 0, which means the stem cell population disappears. But now if I add treatment to that, it still recapitulates our data to some extent. So we're sort of killing off non-stem cells, but we have this nonlinear relationship to stem because we're sort of feeding the stem population by that transition back into stem, right? And by these two parameters, what we see is we see a deeper response for the BCR-ABL1 negative patients than the positive patients. So we sort of recapitulate that disease severity in a different way. In the healthy remission patients, they wouldn't be treated, but if they were, it'd be easy to treat them because it's all the non-resistant cell type, right? And so what I've sort of shown you is through this process, we can sort of recapitulate some intuitive parameters set for BCR-ABL1 positive and negative, and this remission as well. And so I think, yeah, I have a few minutes, right? So what I want to do, this is sort of the conclusion of my talk. I'm happy to answer more questions about this, but since this is a math oncology workshop, I also like to sort of state some statistics about our field at the end of my talks lately as well. So here's a quick update of a couple slides on the Math Oncology website and statistics. So many of you probably know that we run the mathematical-oncology.org website. I'm very happy for any of you to write a blog post or contribute some of your papers to be published in the newsletter. But here's our current level of statistics of where math onco people are in the world and in the US. I don't have any data for Canada, sorry about that. But we are represented in 41 US states and 69 countries, which is really cool. So a lot of Florida here at Moffitt, where I'm at, a lot of California, and some interesting missing states. Maybe we should do some outreach in Math Onco in these sort of gray areas as well. Yeah. So I don't know if you know any colleagues in those states, but you can reach out to them and see what they're working on. The newsletter, we have around 2,000 subscribers and we've done 300 issues. If you go to this website, I don't often mention this, but there's a button to submit your paper. So whenever you publish something relevant, just put it in the Google form there and it will make its way into the newsletter. Don't feel embarrassed about sharing your work. We definitely want to publish everything that's relevant in the newsletter. We also have a blog that is an opportunity for you all to talk about your research more. We've had 88 different authors, which is really cool. And I think it's really generated some interesting discussions as well. And the coolest part is the artwork, right? So we have a cover art featured every week. So if you're artistically inclined, please feel free to submit a cover art based on your work as well. This is driven by an expanding team now, so it's not just me doing all these things. It's really a group of researchers from around the world, so thanks to them. And if you're interested in helping out with this, talk to me afterwards as well. And with that, I'll thank my collaborators for the original ALL project, including my clinical collaborators, Curtis Gravenmeyer, and my postdoc, Sadegh Marsman, who helped with this project. Happy to take any questions and thanks for your attention.
Some quick questions before we head out for lunch.